So imagine you're pulling up to a massive grain silo in the American Midwest.
Right, right.
The air is just, thick with the smell of dry earth and diesel fuel.
Classic harvest season.
Exactly. But you're standing there watching federal agents literally force a company to buy up and physically destroy 500,000 bushels of soybeans.
Wow.
We are talking about $3.5 million of perfectly good food, just completely wiped out.
Just gone.
And why? Because the year before, in that very same field, someone planted a few experimental stalks of corn that were secretly engineered to grow a vaccine for pigs.
Yeah, the Protogene incident. It's
Yeah.
a staggering image, isn't it?
It's wild. A few leftover seeds sprouted in the wrong field, and suddenly the human food supply is contaminated with veterinary pharmaceuticals.
It really gets to the heart of, well, the illusion of control. I mean, we like to think of agricultural genetics as precise, right? Like writing code on a computer.
Yeah, you delete a line here, paste a line there.
Exactly. You get exactly what you want. But biological systems aren't silicon. They are, they're messy, they're chaotic, and they are just relentlessly opportunistic.
Which is exactly what we're exploring today. So welcome to our deep dive into the source material.
Glad to be here.
Today, we are looking at this highly detailed, incredibly rigorous response to a USDA request for information, or an RFI.
Right. And the central question the USDA is asking the public and really the scientific community is this. Do genetically engineered organisms, so GE organisms, do they present materially different plant pest risks compared to conventionally bred organisms?
And based on that answer, should the USDA shift their regulation from their current kind of stricter framework, which is 7 CFR Part 340, over to a much looser framework, Part 330?
That's the core of it.
Okay, let's unpack this right away, because if you're listening to this while driving or, making coffee, Part 340 versus Part 330 sounds like the most boring, bureaucratic alphabet soup imaginable.
It really does.
But why does a shift in regulatory codes matter to you? I mean, it matters because this seemingly dry legal debate dictates the fundamental rules of what ends up in your local grocery store.
Exactly.
What blows into your neighborhood park,
Yeah.
and what permanently alters the global food web.
Yeah, the stakes are incredibly high.
So our mission today is to tear into this massive response document. And the authors of this paper argue, forcefully, and with an absolute avalanche of peer-reviewed data, that GE organisms absolutely present entirely novel risks.
Novel risks that conventional breeding could never, ever produce.
Never.
Right. If we step back and look at the bigger picture here, we are looking at a fundamental, really an existential debate about biology.
Yeah.
Is biotechnology just a slightly faster, more high-tech extension of traditional farming? Or is it a completely new frontier that requires entirely new safety paradigms?
And the document we're analyzing argues that treating a GE plant the same as a conventional plant is, well, a catastrophic failure of logic.
It is. They do not pull their punches.
And it is going to be a wild ride. We are going to explore the underlying biology of genetic engineering, like why a gene isn't just a gene.
Right.
We're going to look at those jaw-dropping, documented case studies of containment failures. We are going to dive into this invisible world of microbes that can literally turn soil toxic.
That part is terrifying.
Oh, absolutely. And we're going to talk about horizontal gene transfer, which, spoiler alert, actually involves electric eels acting as living flash drives.
Unbelievable stuff.
And finally, we will look at the massive regulatory overhaul these authors are proposing.
It is a comprehensive look at the unintended consequences of human ingenuity.
So let's start with the biology, because the USDA's premise, the whole reason they issued this RFI, is that they claim their experience over the last few decades just hasn't identified any, quote, meaningful differences in risk profiles between conventional organisms and GE organisms.
They're basically saying a tomato is a tomato.
Right, which the authors point out is fundamentally biologically flawed.
Extremely flawed. To understand why, you really have to look at the mechanisms of how these genetic changes are actually made.
Okay, walk me through it.
Let's look at conventional breeding first. This is the kind of agriculture humans have been doing for thousands of years. It relies on, well, sexually compatible plants.
Right.
You are moving genes between organisms that could theoretically naturally interbreed, and the mechanism driving this is called homologous recombination.
Okay, hold on. Break that down for me. If I'm a farmer and I have a really tasty tomato, but it bruises easily, and then I have a tough tomato that tastes like cardboard, I cross-pollinate them.
Right.
and their
Exactly.
chromosomes line up and they swap some native traits,
Yeah.
but they are both still tomatoes. They're speaking the same biological language.
Precisely. It's a natural pairing of chromosomes.
Mm.
The plant's native regulatory systems, like the biological software that tells genes when to turn on and off, they remain largely intact and functional.
Okay, I'm with you.
But genetic engineering is an entirely different universe. It is not constrained by sexual compatibility at all.
Wow, okay.
In a lab, you can take genes from viruses, from deep-sea bacteria, from insects, animals, or even entirely synthetic sequences designed on a computer, and just force them into a plant's genome.
Force them in how? Like
It-
with a tiny syringe or something.
Not quite. The insertion mechanisms themselves are actually highly invasive. The document lists several, including transfection, biolistics, CRISPR, and agrobacterium.
Wait, hold on. You just casually dropped the word agrobacterium?
I did, yeah.
Are you saying scientists are intentionally using a bacterial infection as, like, a delivery truck for plant DNA?
Mm-hmm.
How does that even work at a microscopic level?
That is exactly what they're doing, and it's fascinating. So Agrobacterium tumefaciens is a soil bacterium that naturally causes crown gall disease in plants.
Okay.
It does this by essentially hacking the plant. The bacterium carries this tiny ring of DNA called a T plasmid.
Right.
When it infects a plant, it injects a piece of this plasmid, the tDNA, directly into the plant's own genome, basically forcing the plant to produce food for the bacteria. Oh, wow. So it's literally a natural genetic hacker.
Exactly. So genetic engineers essentially hijack the hacker.
Oh, that's clever.
They hollow out the disease-causing genes from the tDNA and replace them with the desired trait.
Like herbicide resistance or whatever?
Yeah, or an insect toxin. Then they just let the bacterium infect the plant cells in a petri dish, delivering the new payload.
That sounds incredibly complex. What about the other one you mentioned, biolistics?
Biolistics is literally a gene gun.
A gene gun.
Yeah, they coat microscopic particles of gold or tungsten with the engineered DNA, and they shoot them at high velocity directly into plant tissue.
You are kidding.
No, hoping the particles just punch through the cell walls and the DNA integrates into the nucleus.
So we are shooting gold bullets coated in synthetic DNA into cells. I mean, that doesn't exactly sound like two tomatoes getting cozy in a greenhouse.
It isn't. And the authors argue this introduces novel risks that simply do not exist in conventional agriculture.
Like what?
Well, the first major risk they identify is viral recombination. What does that mean in this context?
So many genetically engineered plants are designed to be resistant to specific plant viruses, right?
Okay, sure.
To achieve this, scientists often insert genetic sequences from the virus itself into the plant's DNA. It's essentially teaching the plant's immune system to recognize it.
Like a vaccine.
Sort of, yeah. But these inserted viral sequences are what we call heterologous sequences.
Meaning they don't naturally belong in that plant's genome.
Right. So the danger is that out in the field, these plants are constantly exposed to natural wild viruses.
Yeah.
When a wild virus infects a GE plant that is churning out engineered viral proteins, there is a documented risk of recombination.
Right, really?
Yes. The wild virus can actually swap genetic material with the engineered viral genes residing in the plant.
Let me guess.
Yeah.
We are accidentally building better viruses.
Yes. The document cites studies showing this can create entirely new, novel viral strains that could potentially have expanded host ranges or increased virulence.
They could become superviruses.
Exactly. And conventional plants just don't carry those isolated engineered viral sequences, so that specific recombination pathway simply doesn't exist for them.
Wow. Okay, so creating a novel agricultural plague by accident. What else?
There is also horizontal gene transfer, or HGT. We will dive deep into this later, but in short, GE plants often contain antibiotic resistance genes.
Wait, why would a plant need to be resistant to antibiotics? I mean, plants don't get bacterial infections in the same way we do, right?
Right. It's not for the plant's benefit in the field. It's actually a tool for the scientists in the lab.
How so?
When you use that gene gun or that agrobacterium to insert a trait, it doesn't work on every single cell. It's actually a very low efficiency process.
Okay.
So they attach a marker gene to the desired trait, and usually this marker is an antibiotic resistance gene.
Let me see if I follow. After they shoot the cells with the gene gun, they literally dump antibiotics on the whole petri dish.
You've got it.
The cells that didn't take the new DNA die. The ones that survive are the ones that successfully integrated the new trait, along with the antibiotic resistance.
You've got it perfectly. It's a selection mechanism. But the problem is that antibiotic resistance gene stays in the plant's DNA forever.
Right.
It's in the roots, the leaves, the pollen, And the authors point out a severe risk. When that plant material decays in the soil, those resistance genes could potentially transfer horizontally into soil microbes.
Meaning the soil bacteria just pick up the loose DNA and suddenly become immune to antibiotics.
Potentially, yes. Again, conventional plants don't have these synthetic antibiotic resistance markers, so they don't pose this risk.
Right, but let me push back a little here on the core biology.
Sure.
Because the document talks a lot about unintended gene activation and promoters.
Right.
Isn't a gene just a string of biological code? Why does it matter if we drop a new gene into a plant? If the plant looks normal, grows normal, and produces food, does the exact location of the insertion really matter that much?
That is a brilliant question, and it's where the USDA's logic really falls apart, according to this document.
Okay.
What's fascinating here is that the context of a gene matters just as much as the code itself. Think about a promoter as an on switch, or like a volume dial for a gene.
Okay, a volume dial. I can picture that.
When genetic engineers insert a new trait, say a toxin to kill caterpillars, they want to make absolutely sure the plant actually produces that toxin in high amounts.
Right, they want it to work.
So they attach a very strong, highly active promoter sequence to the front of the gene. A commonly used one is actually from the cauliflower mosaic virus. It forces the gene to express continuously.
So it cranks the volume to maximal.
Yes. But here is the critical flaw. Whether you use agrobacterium or a gene gun, the insertion site in the plant's massive genome is often completely random.
Wait, really? So it's like putting a megaphone in a quiet library, but you're tossing it blindly into the dark.
That is a perfect analogy. You drop a megaphone into a random aisle of a massive library. The megaphone doesn't just amplify the person holding it.
Oh, man.
It amplifies everyone standing nearby. If that strong viral promoter lands right next to a naturally dormant cryptic gene in the plant's native DNA, it can accidentally turn that dormant gene on.
And what do these dormant genes do?
Well, they can do anything. Plants have massive genomes full of ancient, deactivated evolutionary traits.
Like genetic ghosts.
Yes. That accidentally amplified gene could produce a novel allergen, a cellular toxin, or even an enzyme that degrades the plant's own nutritional value.
Wow.
Conventional breeding relies on the plant's highly regulated native promoters. It doesn't randomly insert viral megaphones.
That makes total sense. You aren't just adding one new trait. You might be waking up ghost traits that have been asleep for millions of years.
Precisely. And there are other profound biological differences. Some GE plants use RNA interference, or RNAi.
Okay.
These are engineered artificial RNA molecules designed to silence specific genes in a pest that eats the plant.
Like a genetic silencer.
Exactly. It turns off the pest's ability to survive. But the authors highlight that these RNAi molecules can have severe off-target effects.
Like hitting the wrong bugs.
Yes, they might inadvertently silence completely different genes in beneficial insects like bees or ladybugs or even in soil organisms.
How concerning.
And then there is the issue of continuous toxin expression.
This is the bee-strat crops, right?
Yes. Crops engineered with genes from the bacterium Bacillus thuringiensis, or BT.
Right.
Now, in conventional or organic agriculture, a farmer might spray a natural beet biopesticide on a crop to deal with an outbreak.
Right, I've seen that. It's topical. You just spray it on the leaves.
Right, and it's temporary, washes off in the rain, and it degrades rapidly in the ultraviolet light of the sun.
Okay.
But a GEBT crop is engineered to express that insecticidal toxin internally, continuously, 24 hours a day, 7 days a week, in all its tissues.
Wait, even the parts the bugs don't eat?
Yes. The roots, the stem, the pollen, the nectar. It creates an environmental exposure profile that is mathematically impossible in conventional farming. You are turning the entire biology of the plant into a permanent pesticide factory.
Okay, so that's the microscopic theory. The biology clearly has completely different risk vectors.
Absolutely.
But theory is one thing. What happens when these engineered traits leave the sterile laboratory, bypass the safety protocols, and enter the absolute chaos of the real dirt?
Right.
Because the document provides some terrifying examples to back up their claims. Let's look at the documented failures. The authors call them out specifically to prove these aren't just hypothetical laboratory fears.
No, these are documented case studies of agricultural containment failures. And they are critical to the author's argument because they demonstrate that the regulatory framework routinely fails to account for human error and, frankly, the relentless persistence of nature.
Okay, let's go back to the story I opened with. Protogene. The year is 2002. Explain to me exactly how pharmaceutical corn ended up in a soybean silo.
Sure. So Prodigy was experimenting with what are known as pharma crops. They genetically engineered corn, not for food and not for animal feed, but to function as a biological bioreactor to produce an experimental vaccine for a disease that affects pigs.
Which, I mean, on paper, sounds very innovative. Cheaper than a massive stainless steel laboratory bioreactor, right?
Oh, very cheap. So they grew this pharma corn in experimental open-air plots in Iowa and Nebraska.
Open air.
Yes, they harvested their test crop and supposedly cleaned the field. But the following year in Nebraska, the farmer rotated that same field to soybeans, as is common practice.
A completely normal agricultural cycle.
Yes. But some of the seeds from the pharma corn from the previous year had fallen to the ground and survived the winter. They volunteered.
Meaning they sprouted up as weeds right in the middle of the soybean crop.
Exactly. They germinated and grew. And despite federal warnings to the company to identify and completely remove this rogue corn, some of these GE pharma plants were harvested right alongside the conventional soybeans.
And it all goes into the same combine harvester, into the same truck, and into the same grain elevator.
Yes. It mixed with more than 500,000 bushels of stored soybeans. The USDA had to step in, quarantine the entire facility, and mandate the physical destruction of the entire batch.
Wow.
The company was fined $250,000 and had to cover the $3.5 million cost of the destroyed beans.
I was thinking about this earlier. It's almost worse than an oil spill.
How so?
Well, if a pipeline bursts and you spill oil in a field, it's a disaster, but it's static. The oil just sits there. You dig up the dirt, you clean it up, and the spill is over.
Right.
But if you spill genetically modified seeds, they multiply. It's an oil spill that actively manufactures more oil every single spring.
That is a phenomenal way to frame it. Biological pollution is inherently self-replicating.
Yeah.
And the authors use this to point out that the conventional comparator here is absolute zero. No conventional corn naturally produces experimental pig vaccines.
Right.
A conventional volunteer corn plant in a soybean field is just an annoying weed.
Mm-hmm.
It doesn't threaten to inadvertently dose the human food supply with veterinary biologics.
Which brings us to a case where the human food supply actually was contaminated.
Yes.
Starlink corn in the year 2000. Now, I vaguely remember this making the evening news. I always assumed it was just a bad batch of corn that got moldy or something.
Oh, it was far more systemic than that. Starlink is a classic textbook case of massive supply chain contamination.
Okay, what happened?
Starlink was a GE corn engineered by Aventis to produce an insecticidal beet toxin called Cry9C.
Okay, so a built-in pesticide.
Right. But during the regulatory review, scientists noticed that this specific Cry9C protein had two very concerning characteristics that are shared by almost all known human allergens.
Oh.
First, it was highly heat stable, meaning cooking it wouldn't destroy the protein's structure.
Right.
Second, it was highly resistant to digestion by gastric fluids. It wouldn't break down easily in a human stomach.
So if you're allergic to it, your body has plenty of time to react to it, and cooking the food doesn't protect you at all.
Exactly. Because of these glaring red flags regarding potential human allergenicity, the EPA actually made a very unusual decision.
What did they do?
They approved Starlink corn, but only for animal feed and industrial use. They explicitly banned it from the human food supply.
Only for animal feed. Which, when you were talking about tens of thousands of acres of corn and pollen that literally blows in the Midwest wind, sounds like an incredibly naive boundary.
It was an illusion of containment. The regulations required a 660-foot buffer zone around the Starlink fields to prevent cross-pollination with food-grade corn.
Right.
But corn pollen is incredibly light. It drifts on the wind. The buffer zones failed spectacularly.
Of course they did.
The Starlink pollen drifted into neighboring fields of conventional corn destined for human consumption and fertilized the silks.
So the neighboring farmer, who didn't buy Starlink, didn't want Starlink, and thought he was growing normal food corn, was suddenly harvesting grain that contained the allergenic Cry9C protein.
Yes. And because the commodity grain system is just this massive funnel where crops from hundreds of farms are mixed in local elevators, then mixed again on trains, then mixed again at processing plants, the supply chain completely commingled.
Wow.
Within just 1 year of its discovery in the food supply, this unapproved, potentially allergenic protein was detected in nearly 10% of all U.S. grain tests.
10% of the entire U.S. grain supply. from a product that was explicitly banned for human consumption.
It ended up in supermarket taco shells and triggered a massive multi-million-dollar recall of human food products.
I remember that now.
Hundreds of people reported severe allergic reactions, though definitive clinical links were difficult to establish post-market. But the economic damage was already done.
Again, the authors of this RFI response are pointing out that this risk is unique to GE.
Exactly. Conventional topical beet sprays degrade rapidly in sunlight and heat. They aren't heat-stable systemic proteins locked inside the grain kernel itself. The absolute inability to recall a genetic trait once it escapes via windblown pollen is a fundamentally novel risk.
Okay, the third case study they present here is fascinating because it's not a pharmaceutical and it's not a built-in pesticide. It's Enogen corn.
Enogen corn was developed by Syngenta. It's a yellow field corn engineered specifically for the biofuel industry.
Wow.
It produces a specialized, highly active, heat-stable alpha amylase enzyme right inside the kernel.
What does that enzyme actually do?
So in ethanol production, you have to break down the complex starches in the corn into simple sugars so the yeast can ferment them into alcohol.
Right.
Usually, factories have to buy this enzyme as a liquid and add it to the mash. Enogen corn grows the enzyme itself. It makes the ethanol process much more efficient and cheaper.
Okay, so let me guess the disaster here. The Enogen corn cross-pollinated with neighboring food corn, and the new enzyme caused the neighboring corn to rot in the fields because it couldn't handle the genetic change.
Actually, no. And that is what makes this case so insidious. The neighboring corn didn't die. It grew beautifully. The living plant was perfectly healthy. The problem occurred in Nebraska, which is the epicenter of food-grade white corn production in the U.S.
Wait, there's a difference between white corn and yellow corn besides just the color.
A massive economic difference.
Yeah?
This white corn is grown under very strict contracts for premium prices. It's used to make high-quality tortillas, tortilla chips, and hominy. Its starch profile has to be absolutely pristine.
So what happened when the Enogen corn pollen blew into the white corn fields?
The document cites a rigorous, peer-reviewed study from 2024 that mapped this exact scenario. The researchers used a Nelder wheel experimental design.
An elder wheel? What is that?
It's a circular planting pattern, like a giant target. You put the source plant, the Enogen corn, in the very center, the bullseye.
Okay.
Then you plant the white corn in concentric circles radiating outward in all directions. It allows you to perfectly map how far the wind-blown pollen travels and fertilizes the surrounding plants, regardless of wind direction.
Oh, that's a clever way to test a buffer zone. What was the required buffer zone for Enogen?
Syngenta stewardship guidelines recommended a mere 9-meter buffer zone. That's less than 30 feet.
That's nothing.
But the Nelder wheel study proved that Enogen pollen was traveling and successfully cross-pollinating the white corn up to 70 meters away.
Wow.
And their exponential decay models indicated it likely traveled significantly further than that under normal weather conditions.
So the white corn gets fertilized by the Enogen pollen. It grows a cob. The farmer harvests it. It looks like normal corn. When does the disaster strike?
When it hits the factory.
Oh, no.
The cross-pollination creates scattered yellow kernels on the white cobs, and those kernels carry the alpha amylase trait. When that contaminated white corn is harvested, shipped to a food processor, ground into masa flour, and mixed with hot water to make tortilla dough.
Oh, no. The heat activates the enzyme.
Exactly. The enzyme wakes up and does exactly what it was engineered to do. It rapidly breaks down the complex starches into simple sugars.
The dough.
The premium firm tortilla dough suddenly liquefies into a sticky, syrupy, useless mess. It literally melts on the factory machinery.
This blows my mind because the Enogen corn isn't harming the living white corn plant at all. It's not a pest in the traditional sense of like a locust or a blight.
Right.
It's a delayed reaction supply chain contaminant.
Yeah.
It completely destroys the commercial and functional value of the processed product. You don't even know your crop is ruined until the factory tries to bake with it.
And this profoundly challenges the USDA's regulatory definitions. The Plant Protection Act technically includes damage to processed products of plants, but the regulatory framework, particularly Part 330, struggles to handle pure economic and functional harm caused by an invisible, wind-blown, delayed reaction enzyme.
Yeah.
The authors argue this is a textbook example of a materially different risk profile that traditional regulation is just blind to.
So we have pig vaccines mixed into soybeans, allergenic insect toxins baked into taco shells, and ethanol enzymes melting tortilla dough.
Yeah.
But as crazy as those are, these are all agricultural commodities. These are managed fields that get harvested and tilled every year.
Right.
What happens when these engineered traits escape into wild, completely unmanaged ecosystems where there is no farmer to pull the weeds?
That is a critical transition, and it brings us to the cases of wild escapes. The document highlights two particularly alarming examples that illustrate the impossibility of biological containment in the wild.
Go.
GE creeping bent grass and epicite corn.
Let's do the grass first. Because to me, grass is everywhere. Grass is the ultimate survivor.
Yeah.
You cannot contain grass.
You really can, and that's the core of the problem. Creeping bent grass is a perennial, wind-pollinated grass commonly used on golf courses.
Yeah.
It is incredibly robust, and frankly, in many natural environments outside a managed golf course, it's already considered a highly invasive weed.
So what did they engineer it to do?
A company engineered a version of it to be resistant to the herbicide glyphosate, which is the active ingredient in Roundup.
Okay, the logic is clear. You plant this on your golf course, you spray the whole fairway with Roundup, it kills all the dandelions and crabgrass, and your GE bent grass survives perfectly, looking like a green carpet.
Precisely. But here is the biological reality of bent grass. Its pollen is incredibly lightweight and aerodynamically suited for long-distance travel.
Uh-oh.
In field trials designed to monitor gene flow, researchers documented this GE bent grass pollen dispersing over 21 kilometers from the test site.
Wait, 21 kilometers. Are you serious?
Yes.
If pollen easily travels 21 kilometers on a breezy day, does the concept of a regulatory buffer zone even exist anymore? It's a complete joke.
It effectively doesn't exist for wind-pollinated outcrossers, and the biological math of this escape is terrifying.
How so?
The authors explain that conventional creeping bent grass is a weed, yes, but if it invades a sensitive riparian zone, land managers can at least control it by spraying glyphosate. But now you have a glyphosate-resistant version. And remember, conventional breeding could never make bent grass glyphosate-resistant, because that trait simply doesn't exist anywhere in the bent grass gene pool.
Right, it was artificially inserted via genetic engineering.
Yes, and because bent grass is a perennial, it's not like corn that dies at the end of the season. It lives year after year, spreading its roots, spreading its pollen.
It just keeps going.
Exactly. It persists indefinitely. It vegetatively reproduces via stolons, and it successfully outcrosses with wild, sexually compatible grass relatives.
Wow.
If this GE trait establishes deeply in natural ecosystems, which evidence suggests it has in certain areas, the primary chemical tool conservationists use to manage invasive grasses is entirely useless. We have manufactured an uncontrollable superweed that conventional breeding could never have produced.
Okay, and then there's the epicite corn. This one sounds like something out of a dystopian science fiction thriller.
It really does, and it underscores the profound ethical and safety boundaries of open-air biotechnology.
What is it?
Epicite was a pharmaceutical company in the early 2000s that engineered corn to produce humanized monoclonal antibodies against human sperm.
A contraceptive corn.
Yes, intended to be processed into a topical gel for anti-fertility applications. Now, it's important to note that this specific crop was never fully commercialized.
Okay, good in.
But the authors of the RFI response include it to illustrate a massive, glaring vulnerability in the regulatory logic.
Connect the dots for me.
Think back to the Starlink or Protogene cases we just discussed, where buffer zones completely failed and volunteers sprouted the next year, commingling the supply chain.
Right.
If an open field commodity crop that is producing medically active, human therapeutic antibodies accidentally mixes with the food supply,
Then consumers are unknowingly exposed to unauthorized biological agents. It's not just an allergy risk anymore, like Starlink. It's involuntary mass medication.
Exactly.
If that corn ends up in the tortilla supply, you are quite literally eating a contraceptive.
Exactly. And conventional agriculture simply does not present this risk vector. No conventional corn naturally produces human therapeutic antibodies.
Right.
Historically, we produce pharmaceuticals in highly contained, sterile, stainless steel fermenters inside laboratories, not in open, dusty fields where birds eat the seeds, wind blows the pollen, and tractors carry mud and kernels from field to field.
Yeah, that's just wild.
The authors argue that applying traditional agricultural regulations to what are essentially open-air pharmaceutical bioreactors is fundamentally reckless.
It is terrifying.
Right.
But as scary as wind-blown pollen and perennial grasses are, plants are mostly stationary. I mean, they're literally rooted to the ground. They take months to grow.
Right.
What keeps me up at night isn't the corn itself. It's what happens when we apply this same genetic engineering logic to things we can't see. Things that multiply by the billions in a matter of hours.
Which brings us to the invisible world. The document spends a significant amount of time detailing the extreme unpredictability of microorganisms, bacteria, and viruses. When you start engineering microbes, the chaotic variables go off the charts.
Walk me through the cases they highlight here.
The first one involves a bacterium called Klebsiella planticola.
Okay, set the scene for us. What is this bacteria?
The parent non-engineered Klebsiella planticola is an entirely ubiquitous, extremely common soil bacterium.
Okay.
It lives naturally in the root systems of almost every terrestrial plant on Earth. It's completely harmless. It just hangs out in the soil, consuming the natural sugars, and exudates that plant roots release.
So a totally normal microscopic neighbor, just doing its thing in the dirt.
Right. In the early 1990s, a European biotechnology company engineered a version of this bacterium. They inserted specific genes to give it a new metabolic pathway, the ability to produce alcohol.
Why on earth would you want a soil bacterium to brew alcohol?
The idea was actually brilliant on paper, intended to solve a major agricultural waste problem.
Really?
Yeah. The concept was to take harvest, waste-like corn stalks, and wheat chaff, put them in a giant vat with this engineered bacterium, let the bacteria ferment the plant matter into ethanol fuel,
Oh, nice.
and then take the leftover nutrient-rich bacterial sludge and spread it back onto the farm fields as a fertilizer.
That sounds like a perfect closed-loop recycling system. Clean energy and cheap fertilizer.
It does. And the company tested the engineered bacterium in sterile soil in their laboratories. According to their tests, it grew fine, and there were no apparent negative effects.
Okay, so what went wrong?
Well, an independent soil ecologist named Dr. Elaine Aim decided to test it. And critically, she didn't use sterile, dead soil. She used realistic soil microcosms.
Wait, what exactly is a soil microcosm? How is it different from sterile soil?
Sterile soil in a lab has been baked or autoclaved to kill everything in it. It's just inert minerals.
Right.
A soil microcosm is a living ecosystem contained in a tube. It contains real dirt taken directly from the environment, which is teeming with a complex web of life.
Like what?
Thousands of species of natural fungi, nematodes, protozoa, and native bacteria all competing and interacting. Dr. Ingham set up these living microcosms and planted wheat seedlings in them.
Okay, so she is testing the bacteria in the real-world thunderdome of living dirt.
Exactly. She had 3 groups. A control group with just water, a group inoculated with the normal wild-type parent bacterium, and a group inoculated with the engineered alcohol-producing bacterium.
And what happened? Did the engineered bacteria help the wheat grow?
In the control group and the parent bacterium group, the wheat plants grew beautifully, developing deep roots and green shoots.
And the engineered group?
In the group with the engineered bacterium, every single wheat plant died. They
Wow.
turned yellow, wilted, collapsed, and died completely.
Why? What went wrong?
Because the engineered bacterium did exactly what it was programmed to do. It produced alcohol.
In the soil.
Yes. It successfully colonized the root systems of the living wheat. It consumed the sugars the roots were naturally producing, and it relentlessly pumped out ethanol right into the root tissue. The lethal threshold for alcohol on terrestrial plant roots is about 1 part per million.
And how much was the bacteria producing?
The engineered bacterium was producing 17 parts per million.
It literally gave the wheat catastrophic alcohol poisoning from the inside out.
Yes. Now think about the broader ecological implications. This parent organism, Klebsiella planticola, is already ubiquitous in soils globally. It survives perfectly fine out there.
Yeah.
If that fermentation sludge had actually been approved and spread on farm fields, and this engineered alcohol-producing variant had established itself in the ecosystem and outcompeted native strains,
You wouldn't just kill the crops in that one field. It could theoretically spread and colonize the roots of terrestrial plants everywhere, pumping out toxic levels of alcohol across entire forests and agricultural belts.
Exactly. It exposes the absolute hubris of testing a genetically engineered organism in a sterile, isolated lab environment and assuming it will behave the exact same way in the incredibly complex, unpredictable web of a living ecosystem. The conventional parent organism is a harmless neighbor. The engineered version, with just one tiny, well-intentioned metabolic tweak, became a potential biological weapon of mass ecological destruction.
That is deeply unsettling. You mentioned a virus case, too.
Yes, and this one leads directly into what security experts call the dual-use dilemma, the mousepox virus.
Here's where it gets really interesting. Okay, lay it out for me.
In 2001, a team of scientists in Australia were trying to create a viral contraceptive vaccine to humanely control plague-level pest mouse populations.
Again, A seemingly benign, helpful environmental goal.
Right. They took the mousepox virus, which normally only causes a very mild, non-lethal illness in laboratory mice, and they inserted a single naturally occurring gene into it.
Which gene?
The gene for interleukin 4 or IL-4.
Okay, I need an ELI5 on IL-4. What does that do?
IL-4 is a cytokine. It's a natural signaling molecule that immune systems use to communicate.
Right.
The researchers thought that by adding the IL-4 gene to the virus would trigger a stronger antibody response in the mice, which would make their contraceptive vaccine much more effective at causing temporary infertility.
So they were just trying to boost the immune system's reaction, but I'm guessing that's not what happened.
The result was a complete, horrifying shock to the researchers. The engineered virus didn't just boost the antibody response.
What did it?
The continuous, unregulated presence of that IL-4 signaling completely shut down the cell-mediated immune response of the mice. It effectively blinded the mice's ability to fight off viral replication.
Oh my God.
It turned a mild background virus into a hyperlethal pathogen. It killed 100% of the mice within 9 days of infection.
100% lethal from adding one immune-boosting gene.
Yes, and it gets worse. It was highly resistant to existing vaccines.
You're joking.
No. The researchers took a separate group of mice that had been previously vaccinated against normal mousepox and exposed them to the engineered IL-4 version. Even the vaccinated mice died. The inserted IL-4 gene essentially acted as a master key that sabotaged the entire immune defense grid.
So these virologists, who are absolute experts in their field, altered exactly one gene, completely expecting a better vaccine, and accidentally created a hyperlethal, 100% fatal vaccine-resistant supervirus.
Yes. And the researchers themselves were so astonished and terrified by the result that they published it specifically as a warning.
I can imagine.
One of the lead researchers explicitly noted the dual-use dilemma. He pointed out that while they did this in mousepox, the underlying biological mechanism is universal.
Oh, no.
If a bad actor or just a careless researcher put the human version of the IL-4 gene into human smallpox, it would dramatically increase the lethality and potentially bypass our vaccine stockpiles.
It's the ultimate proof that we don't know what we don't know.
Exactly. The current regulatory framework largely assumes that we have the computational and biological models to perfectly predict the emergent properties of a single inserted gene.
But we don't.
The mousepox case proves definitively that we cannot. If this kind of unintended immune suppression happened during the engineering of a plant virus, say, trying to make a crop resistant to a blight, but accidentally engineering a virus that completely suppresses the plant's natural immune response, we could accidentally unleash a hypervirulent crop plague that wipes out global harvests in a single season.
Okay, so we've established that the biology of insertion creates novel risks like silenced genes. We've seen that physical containment fails spectacularly, leading to massive recalls.
Right.
And we've seen that engineering microbes can result in catastrophic, entirely unpredictable emergent properties.
Yes.
But as bad as all that is, the document points out something even wilder. We have been assuming that to spread these engineered genes, the organism itself has to travel, like the pollen has to blow or the bacteria has to divide.
Right, the traditional vectors.
But nature has entirely different mechanisms for moving raw genetic code around, things the USDA hasn't even begun to consider. Let's talk about horizontal gene transfer, and specifically, the electrifying reality of natural electroporation.
This section of the document is truly at the absolute frontier of environmental biology. We briefly touched on horizontal gene transfer, or HGT, earlier, the concept of genetic material transferring directly between different species without reproduction.
Yeah.
For decades, the regulatory assumption was that when a GE plant dies and decays, its DNA degrades almost instantly in the environment.
Like it just breaks down into basic chemical mush.
Right, but modern science has proven that is false. We now study eDNA, or environmental DNA.
Environmental DNA.
When a GE corn stalk rots in a field, or its leaf litter washes into a nearby stream, large fragments of its transgenic DNA persist, flowing freely in the water column and the sediment for weeks or months.
And the question the authors raise is, can other totally unrelated organisms in that stream just absorb that floating DNA?
Precisely. And one of the most shocking mechanisms for that absorption is natural electroporation.
Explain electroporation, because it sounds like something from Frankenstein.
It kind of is. In a modern genetics lab, if scientists want to force foreign DNA into a target cell, they often use a machine called an electroporator.
Okay.
It delivers a very specific high-voltage electrical pulse to the cells. This violent electrical shock temporarily destabilizes the lipid bilayer of the cell membrane, literally blowing microscopic pores open for just a fraction of a second.
Whoa.
While those pores are forced open, the foreign DNA floating in the fluid slips inside the cell. When the current stops, the cell membrane heals and seals the new DNA inside.
Okay, that makes perfect sense in a sterile lab with a $1 million machine. But there aren't electroporators sitting at the bottom of the Mississippi River.
Not human-made ones. But nature has its own high-voltage equipment. The document cites a stunning recent peer-reviewed study demonstrating that electric eels can act as living natural electroporators.
Wait, literally an electric eel. The fish.
Yes. Electric eels, electrophorus, can discharge up to 860 volts of electricity to stun prey or defend themselves.
Okay.
Researchers set up an experiment where they placed zebrafish larvae in a tank of water. They introduced environmental DNA into the water, specifically a plasmid containing a gene that codes for a green fluorescent protein.
So the DNA gets inside the fish literally glows in the dark.
Exactly. They exposed the zebrafish larvae to a natural electrical discharge from an electric eel, and the results were undeniable. The biological electrical pulse opened the cell membranes of the larvae, the floating eDNA rushed in, and 5% of the wild zebrafish larvae successfully integrated the gene and began expressing the green fluorescent protein.
That is absolutely insane. It's like leaving a highly classified encrypted blueprint on a park bench.
Yeah.
We used to arrogantly think no one could read it because it wasn't inside a secure computer. Now we find out a natural force. Literally, a fish swimming by can act like a flash drive, violently downloading that loose blueprint directly into the local wildlife.
It fundamentally rewrites our understanding of genetic containment. And the authors point out it's not just electric eels.
What else?
Massive electrical fields are generated by lightning strikes hitting soil or bodies of water.
Oh, wow, lightning.
Even human activities like electrofishing, where wildlife biologists use wands to pulse electricity into streams to temporarily stun and count fish populations, could act as massive electroporation events if there is transgenic eDNA floating in that water from a nearby GE farm.
So if a GE crop field borders a river and the harvest waste washes into the water, a single lightning strike during a summer storm could theoretically force those engineered pesticide genes or antibiotic resistance markers into the native fish, amphibians, or waterborne bacteria.
That is exactly the risk vector they are demanding the USDA investigate. And the document doesn't stop at electricity. They highlight another incredibly intimate pathway for horizontal gene transfer: oral consumption.
Okay, so eating it. But wait, think about the bowl of GE corn chips you might be snacking on right now. The assumption has always been that the hydrochloric acid in your stomach is a biological incinerator. It destroys everything, including the DNA.
That is the outdated assumption. Current molecular evidence shows that the mammalian gastrointestinal tract does not completely obliterate DNA.
Really?
Significant fragments of DNA survive digestion. They pass through the stomach and enter the intestines.
So if I eat a GE corn chip, fragments of the engineered DNA are surviving inside me.
Yes. And the profound concern is whether these surviving transgenes could transfer horizontally into your gut microbiota.
Oh, man.
Our intestines are densely packed with trillions of bacteria, E. coli, lactobacillus, bifidobacterium.
They are swimming in a souk of whatever we just ate.
Exactly. Remember the antibiotic resistance marker genes we discussed earlier? The ones used to select the successful genetic edits in the lab. If a GE plant contains an antibiotic resistance gene, and that plant is processed into food, and that gene fragment survives your digestion, there is a theoretical pathway where that resistance gene transfers into your natural gut bacteria.
Which means we could literally be cultivating antibiotic-resistant superbugs inside our own intestines just by eating a tortilla chip.
It's possible.
Which could completely compromise vital medical treatments if we get sick down the line.
And the risk extends even further. There is emerging evidence that these transgene fragments can cross the intestinal wall into the bloodstream and potentially cross the placental barrier into fetal tissue in pregnant mammals.
That is terrifying.
The authors are furious because the current regulatory framework requires absolutely zero assessment of these natural electroporation pathways, or these oral HGT pathways, in mammals. They're regulating 21st century biotechnology using 20th century assumptions about DNA degradation.
It feels like we are playing a high-stakes game where the regulators refuse to even acknowledge half the rules. We've talked extensively about the raw genes moving around. But what about the physical proteins and the toxins that those genes produce? How are those substances physically moving through the ecosystem once they leave the plant?
Right.
That brings us to the devastating concept of bioaccumulation.
Bioaccumulation and trophic magnification are ecological principles that the document accuses the USDA of completely ignoring for GE organisms.
What are they exactly?
Bioaccumulation is the idea that a substance builds up in the tissues of a single organism over its lifetime because it absorbs the toxin faster than it can excrete it.
Right, like heavy metals building up in a fit.
Exactly. And trophic magnification means that the concentration of that substance gets exponentially higher as it moves up the food chain. The predator gets a massive dose because it eats dozens of contaminated pre-animals.
Walk me through a specific example of how this plays out with a GE crop.
Let's track a Bt toxin, specifically the Cry1ab insecticidal protein produced by millions of acres of GE corn. In the fall, the corn is harvested, and the remaining stalks, leaves, and pollen are left on the field. The rains come, and this transgenic leaf litter washes into a nearby stream or agricultural drainage ditch.
Okay, the biological trash enters the water.
In the aquatic environment, the Cry1ab protein doesn't just dissolve and disappear, it's highly persistent. It physically adsorbs, meaning it chemically sticks to the surface of the algae, the cyanobacteria, and the decaying organic matter in the water.
So the entire base layer of the aquatic food web is suddenly coated in an engineered insect toxin.
Right. Then, primary consumers, herbivorous aquatic insects like mayfly nymphs or caddisfly larvae, eat the algae. They eat it constantly, accumulating the beet toxin in their bodies over weeks. Then, secondary consumers, like predatory dragonfly nymphs or diving beetles, eat the mayflies. They concentrate the toxin even further in their own tissues.
It's moving up the chain.
Finally, an apex predator of that microecosystem, like a frog or a fish, eats the dragonfly.
That's the trophic magnification. By the time the toxin reaches the frog, it isn't just the ambient trace amount floating in the water. The frog is getting a massive concentrated dose delivered directly into its digestive system by eating the contaminated dragonfly.
Exactly. And the authors highlight a specific, incredibly fascinating and tragic vulnerability here: multi-life stage exposure.
Multi-life stage.
Consider the life cycle of that dragonfly or that frog. They don't just live in one environment.
Right. Amphibians and many insects have complex life cycles. They start in the water and they metamorphose and move to land.
Yes. A frog starts its life as a fully aquatic tadpole. The document explicitly references Gossamer stage 25, which is a critical developmental phase for a free-swimming tadpole where its thyroid gland is developing.
Okay.
At this stage, the tadpole is swimming around, actively eating the algae and detritus that is heavily coated in the bleat toxin. It is accumulating the toxin in its own tissues, which could potentially disrupt its thyroid function.
And thyroid function dictates its ability to successfully metamorphose into an adult frog.
Right. Precisely. It is a critical endocrine window. But let's say it survives. It grows legs, loses its tail, and hops onto land as an adult frog. And what does it eat on land?
It eats flying insects,
Right.
like adult dragonflies.
Yes. It eats adult insects that also bioaccumulated the toxin when they were aquatic nymphs and carried it with them into the terrestrial environment.
Oh, wow.
The frog is getting hit twice. It suffers direct aquatic exposure as a tadpole, and then it suffers trophic dietary exposure as a terrestrial adult eating contaminated prey.
It's an inescapable loop of toxicity.
Yeah.
So what does this actually mean for the broader ecosystem? If a frog eats a contaminated dragonfly and a heron eats the frog, are we looking at a silent, catastrophic buildup of engineered toxins that regulators aren't even testing for?
We are, and this poses an existential threat to endangered species. The document specifically names vulnerable amphibians and insects like the California red-legged frog, the Oregon spotted frog, and the Hines emerald dragonfly.
Right, yeah.
These fragile species live in wetland habitats that are often directly adjacent to massive swaths of GE agriculture in the U.S.
And the USDA isn't requiring tests for this.
Current rules require absolutely zero feeding studies across these complex trophic levels. The regulators might require a short 90-day feeding test on a laboratory rat eating the raw corn.
But that's it?
But no one is requiring a 2-year feeding study on a carnivore that eats herbivores that ate the GE plant material out in the wild. The ecological blind spot is massive.
And let's not forget, humans are apex predators too.
Yeah.
We sit at the very top of this food chain. We eat the fish that swim in those streams. We eat the waterfowl that eat the aquatic plants. Some people eat frog legs.
Right.
We could be bioaccumulating these highly concentrated transgenic proteins ourselves entirely outside the monitored agricultural supply chain.
Exactly. The authors are demanding that the USDA implement mandatory, rigorous bioaccumulation risk assessments. If a novel transgenic protein exhibits trophic magnification, meaning the concentration demonstrably increases as it moves up the food web, they argue it should be outright prohibited from environmental release, period.
Makes sense.
The risks to the broader ecosystem are too immense to justify.
Which brings us to the ultimate climax of this massive document, the regulatory reckoning.
Here we go.
We have journeyed through the biological mechanisms that make genetic engineering inherently different. We've seen the multi-million-dollar field failures of starling corn allergens and enogen tortilla melting enzymes.
Yeah.
We've explored the terrifying, lethal unpredictability of engineered microbes like mousepox, the shocking reality of electric eels acting as living flash drives for DNA, and the silent, deadly food web accumulation of BTOT toxins.
It's a lot.
The evidence presented by these authors is absolutely overwhelming. So when the rubber meets the road, what exactly is this document demanding the USDA do about it?
The authors are drawing a hard line in the sand. They vehemently, aggressively oppose the USDA's proposal to shift the regulation of GE organisms over to 7 CFR Part 330.
Okay, remind us why Part 330 is such a regulatory disaster in their eyes.
Because Part 330 is a generic reactive plant pest rule. It was designed for traditional agriculture. Crucially, it only grants the USDA authority to regulate organisms that are already known and classified to be plant pests. If a new GE organism isn't currently recognized on a list as a pest, Part 330 offers absolutely no statutory authority for a rigorous, precautionary safety review before it is released.
It's a wait-and-see approach.
Exactly. It's completely reactive. And as we've seen with the bent grass and the microbes, by the time an engineered organism proves itself to be a devastating test under the loose definitions of Part 330, it is already deeply established in the environment.
You can't put the biological toothpaste back in the tube.
You cannot. So instead of adopting Part 330, the authors offer the USDA 2 distinct, highly detailed paths to reform their current framework, Part 340. Option 1 is the maximalist approach. We can call it the radical ban.
I love the sound of radical ban. Let's hear it.
Option 1 demands a complete, unequivocal, and total ban on the outdoor release of all GE organisms. No open-air field trials, no commercial cultivation, no exceptions.
Wow. Total ban.
They argue that the documented history of containment failures proves definitively that it is physically and biologically impossible to control these organisms once they are outside.
So if a biotech company wants to grow GE crops, where do they go?
Under Option 1, all GE organisms would be legally confined to heavily fortified enclosed spaces.
Like a bunker.
We are talking biosafety level 2, or BSL2, containment laboratories, or heavily contained, HEPA-filtered greenhouses with physically sealed drainage systems to prevent any eDNA or microbial escape into the groundwater.
That's intense.
Even the transportation of these organisms would be radically restricted.
Meaning what? A farmer can't just throw a bag of GE seed in the back of a pickup truck and drive down the highway.
Absolutely not. The draft language provided by the authors explicitly bans transport in open bed trucks, trains with uncovered flat cars, or anything covered merely by a canvas tarp.
Because of the wind.
They point out that a tarp is not biological containment against microscopic pollen or microbial spores whipping around at 60 miles per hour. Transport would require fully sealed, locked, tamper-evident secondary containment vehicles, similar to how we transport biohazardous medical waste.
Option 1 sounds like absolute science fiction, locking all our biotechnology agriculture in underground biosafety bunkers. It sounds extreme.
It does.
But honestly, after spending the last hour hearing about electric eels downloading DNA and a single gene tweak creating a 100% lethal mousepox virus, it suddenly sounds a lot more reasonable. It sounds like basic self-preservation.
It is a maximalist interpretation of the precautionary principle. But the authors are pragmatic. They know the USDA and the massive agricultural lobbying groups will aggressively fight option 1.
Oh, for sure.
So they offer option 2, the massive overhaul of Part 340. If the government insists that outdoor release is going to continue, the regulatory requirements must be drastically scientifically strengthened to reflect 21st century biological realities.
What does that overhaul actually look like in practice? Give me the heavy hitters.
It is incredibly comprehensive. First and foremost, they demand mandatory multi-omics testing for every single new GE organism.
Wait, stop right there. Break down multi-omics for me. What exactly are regulators looking at now, and what is this new demand?
Currently, regulators use a very narrow, targeted approach. They look at the specific sequence of the DNA they inserted, and they test to see if the plant is producing the 1 or 2 specific proteins they meant to create.
Okay.
It's like checking to see if you successfully installed the new app on your phone, but ignoring whether the app is secretly draining your battery or deleting your contacts.
They only look for what they expect to find.
Exactly. Multiomics is a suite of ultra-sensitive, holistic molecular analyses that looks at the entire biological system. It starts with transcriptomics.
Which is?
This looks at the complete set of RNA transcripts produced by the genome. It asks, did inserting that viral promoter accidentally turn on silent, cryptic genes elsewhere in the plant?
Looking for the megaphone in the library effect?
Yes. Next is proteomics. This maps every single protein the plant is currently producing, comparing the GE plant to the conventional parent. It asks, are there novel, unexpected, mutated proteins appearing that shouldn't be there?
Because proteins are what actually cause the allergies and the toxicity, like Starlink.
Exactly. Then there's metabolomics. This is the analysis of all the small chemical molecules and metabolic byproducts the plant produces. Is the GE plant suddenly accumulating toxic alkaloids because a metabolic pathway got jammed by the genetic edit?
Right.
And finally, epigenomics. This looks at chemical modifications to the DNA itself, like DNA methylation, that don't change the underlying genetic code, but permanently alter how the DNA folds and functions over generations.
It's literally mapping the entire microscopic reality of the plant, not just the single edit.
And crucially, the authors demand that all of this multiomics testing must be done before and after the application of the intended herbicides or pesticides.
Wait, why does that matter? Why test it after you spray it?
Because spraying a plant with toxic chemicals like glyphosate induces massive physiological stress.
Right.
A GE plant might look perfectly normal in a pristine greenhouse, but when you hit it with herbicide in a field, that stress might trigger the plant to produce different, completely unintended proteins or allergens in response. They want to catch those hidden synergistic effects that only appear under real-world agricultural conditions.
That is rigorous. What else is an option 2? We talked about the frogs getting hit twice. How do they address the bioaccumulation?
They demand strict trophic feeding studies. The current 90-day rodent tests are grossly inadequate. Option 2 requires up to 2-year lifetime feeding studies or the full lifespan of shorter-lived species.
By rats.
And not just on rats. They demand studies on a cascade of organisms. Herbivores that eat the crops, secondary carnivores that eat the herbivores, and even highly sensitive indicator species like parasitoid wasps that lay their eggs inside the herbivores.
They are demanding we map the trophic magnification before it escapes and hits the endangered frogs in the real world.
Right. They also demand extremely strict allergenicity testing using criteria established by the FAO and the World Health Organization.
Okay.
This mandates testing whether any novel protein is heat stable, whether it resists human digestion, or whether its amino acid sequences share structural similarities with any known human allergens, like peanuts or shellfish.
To absolutely prevent another Starlink taco shell disaster.
Okay.
Okay, but what about when things do go wrong? Because history shows they will. What about traceability?
That is a massive, highly contested component of option 2. They demand mandatory public labeling, strict identity preservation, and absolute traceability for all GE organisms through every step of the global supply chain.
Wow.
And they stipulate a crucial detail. This traceability must apply regardless of whether the GE trait is analytically detectable in the final processed product.
So even if the corn has been heavily processed into corn syrup and you can't physically find the DNA with a lab test anymore, the manufacturer still has to trace and label that it came from a GE crop.
Yes, you need an unbroken, audited paper trail from the engineered seed to the consumer's plate. Furthermore, Option 2 completely abolishes the current microbial exemptions.
Yeah, those.
Currently, there are fast-track notifications and regulatory exemptions for what the USDA considers common, well-understood microbes, like certain laboratory strains of E. coli.
But as we learned with the Klebsiella alcohol poisoning, one tiny tweak to a common microbe can make it an ecological weapon.
Exactly. The authors demand those exemptions be universally eliminated. Every single GE organism, macroscopic or microscopic, must undergo rigorous, mandatory permitting and full environmental risk assessments. No fast lanes.
If we synthesize all of these immense demands, from the BSL-2 bunker greenhouses to the multi-omics mapping of proteins to the 2-year wasp feeding studies, what is this document really trying to achieve at a fundamental philosophical level?
Yeah.
Because this isn't just about tweaking a few rules. This is a paradigm shift.
It is a profound call to legally shift the burden of proof. Right now, our regulatory system operates largely on an assumption of safety. An engineered organism is generally presumed safe and permitted for release, unless regulators can definitively prove in advance that it will cause harm. This document is demanding the exact polar opposite.
Okay.
It is the ultimate manifestation of the precautionary principle. It demands that the biotech industry must provide ironclad, multi-generational proof of absolute safety across the entire microscopic and macroscopic ecosystem before a single engineered seed is ever legally allowed to touch the outdoor soil.
It is asking us as a society to finally respect the sheer terrifying complexity of nature. We've gone on quite the journey today. We started by looking at the fundamental biological differences of genetic engineering, how inserting a viral promoter isn't just a simple line of code, but blindly throwing a megaphone into a library and waking up ancient traits.
Right.
We looked at the massive economics bills, the OOPS files, where Starlink corn allergens and Enogen tortilla melting enzymes proved that physical agricultural containment is largely a myth.
We saw how engineered perennial superweeds like creeping bentgrass can spread their pollen 21 kilometers on the wind, entirely outrunning our chemical controls and establishing themselves permanently in the wild.
And then we crossed the boundary into the microscopic world, the terrifying unpredictability of the engineered Klebsiella soil bacteria that accidentally killed wheat crocks with catastrophic alcohol poisoning,
Yeah.
and the well-intentioned researchers who accidentally created a hyper-lethal, immune-suppressing, vaccine-resistant mousepox virus.
Which perfectly highlighted the incredible existential risks of the dual-use dilemma in biotechnology.
And from there, we looked at how nature itself actively rebels against our attempts at containment. The electric eels and the lightning strikes acting as massive natural electroporators, violently downloading loose environmental DNA directly into the cells of wild fish.
Wild.
The big insect toxins washing into our streams, bioaccumulating in the algae, and magnifying up the food web through the double lives of tadpoles and dragonflies, directly threatening endangered species.
All of which culminated in a massive, mathematically supported regulatory demand to completely overhaul the USDA's framework, fiercely rejecting the lenient reactive Part 330 in favor of a fortified, intensely precautionary Part 340.
It brings us right back to that image at the beginning of the show. The federal agents staring at 500,000 bushels of contaminated soybeans. When we try to engineer nature, we arrogantly pretend we are working with a clean, isolated blueprint. But what we actually have is a chaotic, infinitely interconnected web where a single genetic tweak, a single altered cytokine, or a single strong promoter can ripple out into a multi-million-dollar recall, an immune system shutdown, or a toxic cascade through the food chain.
I'd like to leave you with a final thought to really mull over.
Please do.
We discussed how natural forces like the immense electrical discharges of eels or a summer lightning storm can trigger horizontal gene transfer, literally forcing synthetic, laboratory-designed environmental DNA into wild, completely non-target organisms. If our rivers, our soils, our wetlands, and even our own digestive tracts are slowly filling up with these engineered genetic fragments from decaying crops and runoff, and if nature already possesses the electrical and biological tools to force those fragmented genes into entirely new hosts, what unscripted, highly lethal, entirely accidental evolutionary paths are we blindly kicking off right now, out there in the mud and the dark water, far, far beyond the reach of any regulator?
That is a chilling question, and one we all need to be asking every time we walk through the grocery store. Thank you so much for joining us on this deep dive into the source material. It's dense, it's incredibly complex, but it is the invisible, microscopic architecture of our future. Stay curious, Keep looking closely at the food on your plate and keep relentlessly questioning the unseen biological systems shaping our world. We'll see you next time.